are going to start again. And this time we are going to a very practical lecture where we will talk about which button to push and how to set it, and a workflow. By the way, I think it was Kaying who asked, I uploaded now this little quiz that we did together with the answers, so you can have a look in Canvas and find it. So I'm going to share this guy. Yes. Okay, so a typical workflow of how to set imaging parameters for both confocal, like single point confocal, or non-single point confocals, camera-based systems. And this is not set in stone. This is a guideline for you to get started, especially on single point confocals. There's a lot of things that you can change, and the parameters influence each other. So it can be quite puzzling. What do I change first? What is more important, or something like this? So I've set up some sort of workflow to help you. And once you understand really all the parameters, you will understand then what to change. But to start with, it's good to have some sort of guideline. Okay, so let's have a look first at single point confocals, which have so many more parameters to change than camera-based systems. Number one is always the same. Define the scientific question for this particular image that you are about to acquire right now. And in general, you cannot answer all the questions on this planet with one single image. So most of the time, you will have to break up complex questions into several questions to be able to acquire images without compromising too much. Okay, the more answers you want with one image, the more you will have to compromise all over the place, and that is not good. So what do you want to measure in terms of resolution or in terms of time with this particular image that you're going to acquire? Let's say, for example, that you have a big sample and you are looking for the few cells that are positive. And therefore, when you acquire an overview image of your sample, your scientific question, your goal with this image is, I want to find where the cells of interest are. It is not to see the small details of the image. So we have a goal that is clearly, I want to find the cells. So you're setting all the parameters to be able to find the cells, but go as fast as possible. So it doesn't mean that you're going to be able to answer the next scientific question, which will be, now I have found my cells and I want to know the shape of the filopodia, or I say whatever, something very detailed. In that case, you will need to modify all the parameters to be able to answer this new scientific question. So for each image, you may have different scientific questions, and acquiring an overview is not the same thing as acquiring a detailed image. And so you need to reassess these settings all the time. So define your scientific question for this particular image and then set everything accordingly. And the next thing is choose the objective that delivers the resolution that you need. So in the first question, you have defined, I would like, so I have done my overview with very big pixel imaging very fast and everything. And then I have found the cells of interest. And now I'm going to acquire an image that is going to, so this is like a brain section or something like this. And I would like to count the number of dendrites in these neurons. And they are quite crowded. So it's very difficult to identify where each dendrite goes. So what do you do then? It means you have to measure the distance between the dendrites to estimate what resolution you need in the image to be able to separate the dendrites. So you look at your image and you try to figure out the distance. And once you know the distance between the dendrite, the smallest distance that you need to basically resolve, this is the object that will dictate which objective you're going to use. So let's say I want to resolve 500 micron, then I'm going to choose an objective that can deliver this resolution. Okay, so we always start with the scientific question. We figure out what resolution we need, what we're going to measure. And for this, it helps, obviously, to know how you're going to analyze. And this is why when you do a whole round like this, you're going to better understand the requirement for sample preparation and for imaging settings in order to be able to achieve the resolution that is needed. So just always do this round all the way, including the image analysis, so that you understand slowly. You get to understand and the whole loop. Okay, so we have our scientific question. We know how we're going to analyze, and therefore we know what type of distances we need to measure in the image, and we choose an objective that can deliver this. On top of that, of course, we look at an objective that has a required working distance, because sometimes your sample is thick and you have to image inside, and therefore you will need a certain working distance. And then you look at an immersion, an objective with an immersion medium that has the same refraction index as your sample or lower. So if it's mostly cells in a culture dish, you can use a water objective or an air objective. If it's cells embedded in a mounting medium, so if they are in PBS, if they are embedded in a high refraction index mounting medium, you can use a glycer objective. Very high refracting index, you can use an oil objective or a water objective or an air objective. So you can go downwards, okay? Right, so now we have our scientific question. We know the resolution that we need. We have chosen our objective. Next thing we make sure that we have the correct fluorophore combination. So we check that the fluorophore spectra match with the microscope excitation and emission specifications. This is extremely important. We're talking now about collecting photons, and you want to optimize this. So that is the next step. After that, you're going to always set the images in black and white so that you can see all the details regardless of the colors. And you will always have the saturation and underexposure lookup table turned on when you're adjusting all your parameters so that you avoid all saturation and underexposure. So you make sure you have no saturation, no underexposure in or around the areas of interest. Okay, then after that, you're going to choose an imaging strategy. So you have a look at your spectra and you see the excitation and emission characteristics of your microscope, and you say, this is a single point confocal, therefore I have several detectors. Can I image several colors simultaneously, therefore gaining time without having a risk for bleed through? Okay, so you have to choose an imaging strategy. Shall I image sequentially or simultaneously? Can I combine two channels? For example, if I have a sample with 4 fluorophores, can I put the blue and the red together without having a risk for bleed through? Can I put the green and the far red together without having a risk for bleed through? Shall I image spectrally if I have a spectral detector? Okay, so you need to, now you know what I mean when it comes to the hardware, but you need to define a strategy on a single point confocal. And then the aim of all this is to avoid bleed through while optimizing the acquisition time. The next thing that you're going to set is the zoom area. So in a single point confocal, we go point by point, line by line, and therefore we have this amazing flexibility to be able to decide what field of view we're going to image, which part of the sample we can image. Of course, we have a maximum. We cannot image the whole thing at once, maybe, but within what we can do, we could go smaller or bigger. So we can zoom in or not, which means basically we have we scan a smaller area. We can decide which area in the sample we're going to scan. Okay, so we can just zoom, and what we are, how do you adjust the zoom? Basically, the area of interest should fill the image. Do not have like a small object in the middle of the image and then a lot of black around. If you have this type of image, you need to adjust the zoom so that your sample, the area of interest in your sample, fills completely the field of view, okay? Because acquiring black images, it's totally, black pixels, it's totally unnecessary. It takes time. It, fills up your computer. And then on top of that, you're not going to, when you zoom into the image to see the stuff in the center that is interesting, you're going to maybe misjudge the pixel size. So so you have to be careful with your objects if they are moving. Obviously, if you have migrating cells and this is what you want to image, you maybe need to get a little bit bigger field of view so that you allow them to move. After you have chosen the objective, the imaging strategy, and adjusted the zoom to your object of interest, you're going to choose how many pixels you're going to jam into this scan area. Okay, so that will determine the pixel size because we have a certain size that we're going to scan, and then we put a number of spots in it. So obviously the size of the scan divided by the number of spots is going to give us the pixel size, all right? So how do we decide on the pixel density? Very easy, we click on the button. And so when we click on the button, the software is going to have a look at which wavelengths you're using and which objective you're using, and it will calculate the Nyquist or the pixel size that fulfills the Nyquist criterion. Okay, and you push the button, it gives you that. So it will change the pixel size to the correct pixel size. And you could decide to sample more, but you don't need to because it will not give you any more information if you oversample and then if you sample at the Nyquist criterion. You remember this curve that we have. So the Nyquist criterion tells us how many times we need to sample so that we can reliably reproduce the curve. We could put zillions of spots in this curve, but it will not bring us anything. It will just take more time and make more data. So you do not need to oversample. Try to avoid oversampling with a single point confocal. It just goes slower and it bleaches more. On a camera-based system, because we acquire all the pixels at the same time, it doesn't matter. Okay, but on a single point confocal, don't oversample because it takes a lot more time. But so you push the button, you see the Nyquist pixel size, and then you need to ask yourself, do you actually need to use such small pixels to answer your scientific question? So now we go back to point number one, the scientific question, and what do we need to measure? And maybe what the objective delivers might be more than what you need. Okay, so only you need such small pixels only if you need to resolve very small distance at the level of the resolution of the objective. Otherwise, you can actually undersample and be limited by the sampling. So it's okay to undersample and be limited by the sampling as long as you do not need the full resolution that the objective delivers to answer your scientific question. So now we know the pixel size that is required to fulfill the Nyquist criterion. We ask ourselves, do I actually need this? And you can also ask yourself, can you afford to use such small pixels? There are lots of reasons why you may not be able to get such small pixels. For example, if bleaching is a problem, or if you have light samples and you have a light toxicity problem, more pixels, more bleaching, more light toxicity. And or if the imaging is going to take a very long time, and for example, your objective is moving, so you cannot image for so long, etc. So if these things, so you know the Nyquist pixel size, but then you ask yourself, do you need it? Can you actually afford it? What are the consequences of undersampling? So then it's a conscious decision that you make. Why do I pick this pixel size? Why do I decide that I'm going to undersample? Is it okay with my scientific question? And of course, if you undersample, you are now aware that you're going to have aliasing, moire artifacts in your objects. They will definitely be there. And you do not know what the sample looks like. Okay, so if you have, again, it's only the part of the objective resolution, let's say, that is not resolved by the sampling that will create moire artifacts. So normally, if you image very small objects, you will definitely have to go for Nyquist pixel size because you don't want to have moire and you want to use the full resolution of the objective. If you image bigger objects, if you get small objects that create moire artifacts, maybe you don't care. Maybe you may want to consider using a different objective with, for example, less magnification and a bigger field of view. And that would make your imaging faster. And so you would not have to make so many tiles, for example. Okay, so if you undersample for this objective, consider changing objective, maybe that you might have some advantages. So then you have to be aware of the Mori effect, and that you will get some Mori artifacts, and also you can try different degrees of undersampling and compare the images. But it's unlikely that you're going to see the Mori artifacts because our samples are not symmetric and are not like a grid, unfortunately. So it's very difficult to detect. And also because we don't know what they should look like. Okay, so now we have a sample, we have an imaging strategy, we have a sample that is hopefully not, we don't have any bleed through between the channels. And yes, we have a hand up, Hajar. Did you have a question, Hajar?
No, sorry, I just pressed it. I was wondering regarding the optimal, when you said we can change it from the
software.
Does it differ from a software to another? Or it is only...
It differs a little bit. I will talk about this a bit later, but basically they have different names.
Yes.
Number one. Yes. Second, absolutely all companies who make single point confocal will give you an option to automatically set the Nyquist, the pixel size to the Nyquist sampling. That's for sure, absolutely all of them. I don't know the names for all of them. The Zeiss software, the button is called Optimal.
Yes, yeah.
True. And in the Nyquist, in the Nikon software, it's called Nyquist. But I don't know in the Leica software what it's called, but we will get the answer once you actually look for it. I don't remember now who has a Leica Confocal, Kashan. I think you will have to look for it, and then you will tell me what this button is. So ask the person responsible, and then you will know. Maybe someone in the attendee knows what it's called, the button for Olympus and for Leica. Any Olympus and Leica users, single point confocal users out there?
But my next question is, once we pressed it and it gave us like, because sometimes it gives you different numbers than what you start with.
Exactly.
So the right thing to do is to go with whatever we get after pressing the optimal.
So it all depends on your scientific question.
Okay.
So you are going to ask yourself, this is first you're going to calculate, oh, this is the resolution that that this objective will deliver at this wavelength, right? And then you will ask, okay, what are the you do the other way around. You will first have a look, what is the smallest distance that I must resolve in my sample? So if, for example, you need to count, so I will have a little slide about this, but if you need to count crowded nuclei in a tissue, in tissues, in some places the nuclei are going to be very close to each other and very packed, and other places they're very sparse. If they're sparse, you have no problem, because what matters is the distance between the edges. So then, you know, if you have one nucleus here and there, it's very easy to count. You don't need a high resolution. But if you have another place in your tissue where it's very crowded, then this is what limits your counting. You want it to be reliable. And then in that case, you will have to figure out what is the distance between two nuclei and say, okay, this is the distance I need to resolve. So I'm going to pick an objective that does that. And if the distance, if you pick an objective and the maximum resolution of the objective is more or less the distance that you want to resolve, you absolutely need to use the Nyquist sampling pixel size. But if you have the choice between three objectives and one of them does not give you the resolution you need and the other one gives you a resolution that is much, much, much better, then maybe you can undersample, because you do not need that. Then you pick the objective that has the much better resolution, but you do not need to use the full resolution of the objective. Then it's okay to undersample. So it's all about your scientific question.
Yeah, thank you. So yeah, we goes back to the sample again and again.
Everything goes back to the scientific question. So when you push this button, you will see the number of pixels change. And that's because you have defined what scan area you are going to scan. And now you want this to have a certain number of pixels in it so that you can, and a certain number of line so that you actually get the pixel size that fulfills the NIS criterion. Okay?
Yes,
so this interplay is very important between your scientific question, the objective, and the pixel size. Yes, Leonie?
I also have a question.
Yeah.
Because we always calculate the pixel size with the wavelengths that we use.
Yeah.
But usually in my images, I use more than one wavelength.
Yeah.
So
Exactly.
I assume.
Exactly. So you need to basically ask yourself again, okay, I'm going to be imaging some small dots in green, and I'm going to be imaging maybe some nuclei in blue, and some fibers in red, whatever. And you need to ask yourself basically for each of them, what is the requirement for my scientific question? What type of distance for each of these I need to answer? I need to answer reliably my scientific question. And so it might be the case that you will have to use different tracks, as we say, so basically completely different settings and image sequentially and change everything. So maybe you must have the same pixel size for the whole image. So then you go for the wavelength, the scientific question that you have for each wavelength, you will go for the one that is delimiting. So the one that requires the smallest pixel size. This is what you need to use in your image. Okay, so you have to ask for each wavelength, what is the smallest distance that you need to resolve for each different color. And then you go for the smallest one. And you set everything according to that. Okay, so I'm going to try and go a little bit faster because otherwise I won't finish. So here we have, once you have done this, you're going to set the pinhole size. So should the pinhole be optimized for the shortest or longer wavelength? This is a little bit of a tricky one, but basically you remember that the pinhole size, I think I have this, yes, the pinhole size is measured in 1 area unit, and that is because it is normalized basically to the for each wavelength to the distance between the 0 here and the 0 here. So this is the diameter, the radius of the main lobe of the point spread function, and this is the resolution. But in here, the diameter of the main lobe is 1 area unit. Okay, so it's double. It's 1 area unit, and this is what we use when we set the pinhole. In general, we have a button called 1 area unit that we press. Okay, and that closes and opens the pinhole depending on the wavelength, because now we have, for example, this could be the point spread function for blue, and now I also have a far red. So we have a certain size. I have also a far red fluorophore, and the longer wavelength, you remember, longer wavelength, lower resolution. We basically, it's a longer wavelength that we get a bigger point spread function. So maybe the point spread function with the far red is going to look like this. And therefore, if I close my, my pinhole optimally for the blue, I'm going to chop off some of the far red signal, okay? And if I choose my the optimal, if I set the pinhole, we have only one pinhole for everything. So if I set the pinhole to a optimal for the far red, then I'm going to include some of the lobes of the blue, and therefore I will not have the same contrast in my image, in my blue image. So you also need to relate this to your scientific question. Which channel is the most important for you? Which one is brightest? If you acquire, right. So if you want the best contrast for the shorter wavelengths, then you will close the pinhole and the longer wavelengths will be dimmer. If you want the best contrast for the longer wavelengths, then you will open the pinhole to have 1 area unit for the long wavelengths, and therefore you will have a lower contrast for the shorter wavelengths. So it's all related to your scientific question. What is the most important channel? And what is the one in which you have to set the main criteria for, and then you have to compromise for the others, okay? So now we have set the pinholes, And as always, you will try to increase the contrast. This is the role of the pinhole. So if you have very dim samples, by any means, you do not want to add some noise to your image. You know very well now that if you have a low number of photons, you're going to have a high contribution of the shot noise. Therefore, your signal to noise ratio in the image will not be good. That's not what you want. So if you have a dim sample, it's totally okay to open the pinhole more than one area unit. Okay, and you will know that anyway, you will have less contrast in the image, but you will improve the signal to noise ratio. So all this has to be weighed against each other. There is no such thing as setting one area unit for everything. It just doesn't work. So open and close the pinhole, okay, according to your sample. If you have dim samples, open the pinhole a little bit more. And if you have very bright samples, you're very welcome to close the pinhole below one area unit, because that will give you a higher contrast and therefore a more usable resolution. And that is especially true in Z, okay? So we close the pinhole and then we set the laser power. And how do we set the laser power? Well, you have to stay low if you have a problem with bleaching or light toxicity, for example, bleaching over Z stacks or in live samples or something like this. Then you have to keep a low laser power. But if you don't have these problems, just blast the thing. Put a lot of photons on your sample, get very bright images. You will get a low contribution of the shot noise, and bleach those samples. Take images that answer reliably your scientific question, and it's going to be perfect. So and then it doesn't matter if you bleach your sample, you have your images and you will not need to reimage them. So you have to think that way. So don't hesitate to increase the laser power. But make sure that you do not saturate the fluorophores. So you remember, if you have 100 fluorophores in your sample, and once you have excited all of them, you can keep on adding laser power, you're not going to excite more. So what happens then is that they start going into the dark state, and then it looks like your sample is bleaching, but you take another image a bit later and the sample is brighter because they have recovered from the dark state. Okay, so do not send your fluorophores to the dark state, which looks like fluorophore depletion. It looks like bleaching. All right, so now the next point is that, I don't know if I have something else before now. So the next point is that you can choose if you're going to average or not. How to choose if you need to average? You scan continuously. So you take 2-3 scans and you see that the things that change from one image to the next is the random noise. So this noise comes all over the image, but randomly. And you ask yourself, does this random noise prevent you from answering your scientific question? If it does, add some averaging and reassess, and etc. like this. If it does not prevent you from answering your scientific question, don't average. Then you set the detector gain and the offset. And then you set the gain to have no saturation at all, and you can a high gain. Remember that a high gain increases the noise, and therefore it's better to use a slower scan or increase the laser power. This would be your first option if you don't have a problem with high laser power. And set the detector offset to the lowest level that shows no underexposure. You should have no blue pixels. And then finally, you set the pixel dwell time, so they all talk to each other. So you'll probably have to play back and forth a little bit, but again, it's not set in stone. So if all other noise reducing strategies are not sufficient, you can go down in the scan speed. And then you will get more photon to the detector, so less shot noise. But the disadvantage is that you bleach more, you have longer imaging and higher light toxicity, and also higher risk for fluorophore depletion. So in here, I don't know if you can see. Can you guys see the movies playing?
Yes.
Yeah, okay. So you can see here, this is an image without any laser, no sample, no laser, and you can see here the noise. So you have like little dots of color that come a little bit around. And if I now use a high gain, you can see hopefully that we have, sorry, my movies are not playing very well. Anyway, here you would see a lot more dots. So high gain means high noise. This is the readout. So I'm a little bit off target when it comes to the time. So on camera-based system, we have less parameters to set. So again, always the same thing. You define your scientific question for this image, And you, in general, you have to break down complex scientific questions. So define what you want to measure, pick the objective. So this is exactly the same thing that works for you. And then consider the camera size and calculate if the pixel size on the camera fulfill the Nyquist criterion. And then you decide based on your scientific question if it's okay to undersample on the camera side or not, and or depending on the camera, if it does fulfill the Nyquist criterion or not. And then you can add some magnification to actually fulfill the Nyquist criterion, or you can change the objective. Okay, so and if you do not, if you undersample, then you are going to create some worrying artifacts. Then you choose the fluorophore, so it's exactly the same thing. Set the images in black and white, always have the saturation and underexposure lookup table on, and acquire images will have absolutely no saturation. You cannot underexpose with a camera. And you can crop the area, the camera area. If your sample does not fill the whole field of view, you can change objective to magnify more, or you can crop the camera area to not acquire all the pixels. Do not acquire images with a lot of black everywhere. And this is the field stop. I'm just trying to see, oh yeah, I have a few things more. And then this, okay. So I'm almost done. There is something, yes.
Just now when you were saying that we should crop the field,
yeah,
when the sample is not entirely in the image.
Exactly.
I was wondering what you would do, like if you had if you had two objectives, like the 60x and the 100x with the same NA, would you go for the higher magnification, or would you take the 63x and crop the field?
Yeah, so it all depends if your camera fulfills the Nyquist criterion with the 60x or not. I would always go with a lower magnification if I can. So if the camera fulfills the Nyquist criterion with the 60x, if you go to 100x, you're going to have a dimmer image, and yes, you're going to oversample even more. So you don't have much advantage there. So I would go for the smaller one, and I would crop the camera. You get less, so the files will be smaller, and you can acquire much faster if you don't acquire all the pixels.
Right. Okay. But I guess that if you oversample with the 100x objective, then you have less noise, right? Or at least less.
If you oversample with the 100x objective, I don't think so, because the magnification depends also on the numerical aperture. So the magnification plays a big role in the brightness, and and the brightness, if something is dim, then you have more contribution of the shot noise, right? So then the brighter the better when it comes to signal to noise ratio.
Yes. No, I was just thinking with the photons, but I guess you have the same amount of photons.
Yeah, but they are diluted, right? When you magnify more, they go to, so each pixel will receive less photons.
Yeah. Okay.
And therefore you get a higher contribution of the noise. So it is a balance between all these things.
Yes.
If you fulfill the Nyquist criterion with the 60x, I would definitely go for the 60x.
Okay, thanks.
So this is on the excitation side. You remember that I spoke about field stop, and a field stop is basically an iris that is at an image plane. So when you close it, you actually see the edges, and there is always a field stop in the excitation light path. So wherever the excitation light comes in, when we do fluorescence microscopy on a camera-based system, the light can come from a metal light or a lamp, or maybe even lasers, or it can come from LEDs. And when it comes into a microscope, you usually have, you always have some sort of aperture. So it could be something like this. Like this one, FS, is a field stop. This is an Olympus microscope, field stop, and it's a upright microscope. So the excitation light comes from the top, same side as the objectives, right? And so we could close this. And when we close this, we actually can see it in the image because it's a field stop. And this is another microscope from another student. We finally found this. So this is called iris diaphragm in this system. But basically, when you close that thing, what happens is that you prevent the area outside of your field of view from being exposed to the light. You don't need to. If this is the area that you're imaging, you close the field stop to see this, and then you would reopen it just a tiny bit so that you don't see it anymore, but it's just outside the field of view. And that, because maybe if you open it completely, it is very big, and then you bleach the things that you're actually not imaging, which is not very good. So the field stop in the excitation light path is very important also to close and open, to avoid the bleaching outside the field of view. Victoria, I'm sorry, I'm a little bit late, so I will do my best. Just a tiny bit late. Okay, so on the camera, we can also bin. So then you're going to choose if you're going to bin. If the full resolution of the camera is not needed, and where you're heavily oversampling, just bin. You're going to go faster, you'll get brighter images and smaller files. So it's very good if you can do that. Or if you want to do an overview and your scientific question is to quickly find your position of interest, bin your camera. And then we want to decrease the noise, right? And the noise on the camera-based system comes from the shot noise and from the readout noise. So if you have a readout rate option, sometimes they have funny names like optimal or bright or something like this, but these are options that you have that change the readout noise. Then you use the lowest readout noise if you don't need to image fast, because you will have the best signal to noise ratio. But if you want to image fast, then you have to increase the readout. And then, of course, as always, if you can, increase the illumination to decrease the shot noise contribution. And if you have some extra gains, you can also test to try to see what the best signal to noise ratio each gain gives you. The averaging is exactly the same strategy. Scan continuously, see what changes from one image to the next. This is the random noise. Does that prevent you from answering your scientific question? If yes, you can put some averaging. If no, don't. And then background correction on a camera-based system, we always have the camera offset, and that we need to subtract when we want to compare different intensities. And you can also subtract the background and also use shading correction if the illumination is not completely even. So a final last spot. This is Leonie's sample. Do you see any problem with this image? So she has 3 colors. It's the spinal cord section, and the sample is on the slide. Any problem there? It's beautiful, isn't it? I love it. Okay, since I am a little bit squeezed in time, I'm going to give you the answer. Do you see the problem now? Leonie, you're not allowed to answer. Anybody sees the problem? So these are the 3 channels. I've just split the channels and I'm showing them to you in black and white.
I would say that blue and green is like different, difficult to distinguish.
Oh, sorry, because I put the blue image behind the green. I wanted to show you. That's why. But what do you think if you look at the blue and the green?
The blue is not in focus.
Yeah, exactly. The blue is not in focus. Okay, so we take one image with 3 channels. And the red and the green are in focus, but the blue is not in focus. The sample is on the slide. We have prolonged gold. I think it was a prolonged diamond. I don't remember. And I think I wrote it here. Prolonged diamond, which anyway was autofluorescent. And in this case, it was very clear. I think maybe because we had a lot of prolonged diamond. Anyway, it was doing something. There was aberration only on the blue channel. And that is because I am quite sure, but I'm waiting for Leonie to tell me when she re-prepares new samples. I am quite sure that the reason why this channel was out of focus is because the sample is on the slide and not on the cover slip. You will never, and Leonie has said that she has seen this in her sample, and with the next user, I was imaging with the same objective, and we also had 3 different colors, and we didn't have that problem. That definitely came from the sample, and this is a problem of putting the sample on the slide, not on the cover slip, and you will not detect it. You will see something funny and go like, oh, background, whatever, discard the thing. Don't discard these sort of things. You see something strange, address it, okay? This is something that you will not notice until you compare side by side or you have some experience. that's all I wanted to say. So I'm a little bit late, but now sorry about that. I think so let me think.